Hello, everyone. Thanks for coming to our first seminar of the year. Happy 2022. May it be an improvement over 2021 and 2020. But we have two great speakers for you. And before that, I just wanted to remind everyone about the Mycotalk series. The next Mycotalk is coming up on January 27th. It should be two fantastic speakers, Elsa Jacobson and Al Brown, talking about Candida albicans and candidiasis. And so I'll put the info into the chat. It's free to sign up and it's highly recommended. And our first speaker today is Darian Santana. And Darian graduated from Weber State University in Utah in the US with a BS in microbiology. He then worked for Nelson Labs in Salt Lake City, Utah, performing pre-market validation testing for pharmaceutical and medical device manufacturers. He's currently a PhD candidate in Teresa O'Meara's lab at the University of Michigan. And today he'll be speaking about work from his recent publication in Nature Communications on the tools and morphogenesis in Candida auris. So Darian.
Thank you, Jessica, for that very kind introduction. Let's see here. I'd also just like to express my thanks to the organizers for this excellent seminar series. I'm very excited to be a part of it and share some of my recent work with you today. So as Jessica mentioned, I've been talking today about Candida auris. Just as a brief introduction, I'm sure many in this audience do not need much of an introduction to this organism. Candida auris first really showed up on the scene with this report in 2009, where it was isolated from the ear canal of a patient in Japan. And this initial report really sparked surveillance initiatives on a global scale to the point where now we're aware of 4, maybe 5 distinct genetic and geographic lineages of this organism that are responsible for disease and outbreaks on every major inhabited continent. I think recent numbers suggest 44, 45 countries and growing have reported cases and outbreaks of this organism. And that this emergence has been paired with really somewhat enigmatic behavior for this organism. We see widespread antifungal resistance. We see transmission between patients, which is somewhat unique among Candida species. We see elevated levels of thermal tolerance and adaptation to human body temperatures. All of these, beg the questions about what molecular biology is really driving the success of this organism as a human pathogen. And in the O'Meara lab, we like to take a functional genetics approach to addressing some questions like this. And this is something that when I first joined the lab was not a trivial thing to do for Candida auris, simply because the tractability of this organism in performing genetic manipulations was not on par with the development of tools in related fungal species. And that's really where my work picks up. And what I'd like to talk to you a little bit about today is some of the success that we've had in developing new genetic tools that really open up the availability of molecular biology in this organism. And so we can start to ask really critical experimental questions that haven't necessarily been easy to ask before. As sort of a proof of concept for these tools, as I'll share, I'm going to discuss a phenotype that's dear to the heart of many fungal biologists, and that is morphogenesis. So we know, for instance, in Candida albicans, that we see a transition of the cells from a budding yeast stage to hyphal or pseudohyphal cells in response to a number of conditions. It was recognized pretty early on that we don't see that same sort of dimorphic switch in Candida auris for many of the same responses. This is not necessarily to say that we don't see more than one morphology of Candida auris. And there are reports of a subset of Candida auris isolates that have been recovered from patients that form this constitutively aggregating morphology. And these appear to be a failure of daughter cells to separate from mother cells after a bud. The reports of these in the literature that come from case studies tend to show a very high representation for clade 3 and not other genetic clades. And so there has been suggestions that this is a clade specific trend. nonetheless, it's really unknown what is driving this phenotype genetically and what selection may have led to the emergence of this phenotype in a subset of Candida auris isolates. And so we set out as a proof of concept for the development of our genetic tools to answer that very question: what genes in the Candida auris genome might be contributing to an aggregating phenotype? We first took a forward genetics approach to answering this question, just a non-biased screen for genes that would result in the development of an aggregating phenotype in a non-aggregating parental strain. To do this, at the time, there was not, at the time we developed this, there was not a great forward genetic system available in Candida auris. So we turned to the plant pathogen Agrobacterium tumefaciens, which has a proven track record in performing insertional mutagenesis in fungal species. And then just as a brief overview, the way that this works is we have a bacteria that encodes a binary vector system. One of these vectors called the TI plasmid, contains a region known as the transfer DNA or tDNA. And so this region is mobilized by the bacteria into a host plant or fungal cell and integrated into the target genome randomly. And we can engineer this, for instance, to encode a selectable marker for Neisseria thracian resistance, which is what we did for our Candida system. The other plasmid contains an operon with all the genes necessary to facilitate the mobilization of the tDNA. And the important thing is that this is inducible in culture. We can mimic the natural conditions that induce this system to perform insertional mutagenesis in Candida auris. So what I'm showing you right here is a pair of clade 1 Candida auris isolates. These are pretty closely genetically similar, and what we were seeing when we were performing this conjugation is that we had a transformation efficiency of about 1 in every 1,000 cells, which was plenty to generate a pretty sizable mutant collection. Now, as you may have noticed, all of the engineering in this case happens on the bacterial side of the equation, right? So we haven't done anything to prepare the Candida auris cells for the transformation. Because of that, we asked whether we could use the same technique to perform forward mutagenesis in isolates of Candida auris from diverse genetic backgrounds. And in fact, what we saw is that we could do this. And before you say anything, I know some of these bars are a little bit small, but even though we have this wide variability amongst the different clades and different genetic backgrounds, even the smallest bars here are an efficiency that's plenty sufficient to generate a mutant library appropriate for screening. It's just a matter of plating more cells after the transformation, which is not a considerable amount of work. So using the system, we were able to identify morphogenic mutants, those that were aggregating or with a filamentous type morphology, based on a very straightforward macroscopic screen of colony morphology. So as these cells pile on top of each other during division, the colonies that end up forming become wrinkled and dry as compared to the smooth, glossy, wild type colony shapes. And so using the system, we were able to demonstrate that the formation of these aggregating colonies was something that isolates from all 4 major clades, not just clade 3, were capable of doing when probed with the correct transformation efficient mutation with Agrobacterium transformation. So what I'm showing you here is our technique that we had to identify the genes that were disrupted by the transfer DNA. So we used the whole genome sequencing approach to identify the transgene insertion sites. The read maps that I have here are reads that were isolated from so they're Illumina reads that were taken of individual aggregating mutants and then mapped back to the tDNA plasmid from the Agrobacterium strain in the first place. And what you can see is that most of those reads pile up to the transferred transgene region from the tDNA plasmid. But I've highlighted regions in blue and red here that extend beyond the borders of that. And these don't line up to the Agrobacterium plasmid, but instead these reads correspond to the native locus of the transgene insertion site in the Candida auris genome. And so we can leverage this information to identify the transgene insertion sites and determine which genes are contributing to the phenotype. And we can even do this in pools, as I've indicated here. So we've sequenced up to 7 of these mutants at the same time, and it becomes very cost effective when you do this, and very straightforward compared to some alternative approaches for this sort of thing. So I'm showing here a subset of some of our mutant strains that we've done the sequencing to identify the transgene insertion sites. And we noticed a signature, if you will, for genes involved in the regulation of chitin production and degradation. We have chains of cells here with an insertion site in an ortholog of the chitin synthase gene CHS2, as well as insertions here in a pair of members of the RAM pathway. So the RAM pathway is pretty well described in many fungal pathogens as being a regulatory pathway that regulates separation of the daughter cell from the mother cell. So this makes sense. The way that this usually works is through regulation of the transcription factor ACE2, which in turn regulates a number of genes responsible for the degradation of the primary septa between mother and daughter cell, including but not limited to this chitin degradation gene, ortholog like CTS1. And so we identified ACE2 and TAT3 as members of this pathway that showed up in our screen and developed cells that exhibited a similar aggregating morphology to that's been reported with natural isolates in the literature. Because we had these hits, we wanted to follow up on our screening hits with clean gene deletions to see if we could recapitulate and validate these phenotypes. And again, this is something, as I mentioned, that historically has not been a trivial task to perform in Candida auris, which tends to favor illegitimate recombination methods. And so trying to get a deletion cassette into a targeted location can be very labor intensive and very expensive. To address that problem, we adapted a DNA-based transiently expressed Cas9 and guide RNA transformation system, similar to some that have been popularized in Candida albicans and Cryptococcus neoformans for use in Candida auris. And so this system includes two components. So this is just a DNA cassette that's transformed into the cells alongside a deletion cassette. And this cassette encodes a codon-optimized Cas9 that's driven by a Candida auris specific strong promoter, as well as a cassette that drives the expression of an sgRNA to target the Cas9 to the targeted locus. For reasons I won't get into today, we decided to use a polymerase II promoter to drive this. So we've also included some sequence to result in post-transcriptional cleavage sites to deal with the issues that would arise from a 5' cap and a poly A tail on the sgRNA. So we developed these systems. We wanted to test if they actually, improved our ability to get a repair cassette into a targeted site in the Candida auris genome. And so we built a reporter system to assay just that very question. Here's what that reporter system looks like. I have the strongly expressed ENO1 protein in Candida auris, and in this case, the reporter tags that protein with the fluorescent marker RFP. Now this RFP is not driven by its own promoter, so we expected that we would only see robust fluorescence in cells that had inserted the RFP gene precisely in frame to be expressed along with the ENO1 protein. The other side of this reporter cassette is an independently driven selectable marker. Again, we're using the nursery thricin resistance gene NAP. And so what this allows us to do is identify the total number of cells that received the reporter cassette, regardless of whether it ended up in the correct location, right? So here's what that system looks like in practice. I can take a sample of transformants. In this case, I'm showing you 10 representative isolates after a transformation. And I know that they're true transformants because they're resistant to nursery thricin, as I just described. And we can see that 5 out of those 10 in this case are strongly fluorescent. So we would expect that those 5 have integrated the reporter cassette in the correct targeted location. And we can demonstrate that with PCR using primer set A that I've diagrammed out here. And in fact, what we see is that the fluorescent transformants show correct PCR for the targeted integration compared to the non-fluorescent transformants, which do not. So this gives us all the information that we need to measure the rate of a targeted integration event when we perform a transformation. So for this particular isolate, which is AR387, which is a member of clade 1, the South Asian clade of Candida auris, when I just add that reporter cassette by itself in a transformation and electroporate these cells, I'm seeing an integration rate in the correct location sitting around 20%, which is not terrible, but it's also not great. When I add the Cas9 expression cassette as well to the transformation, I don't see a significant increase, which is expected because there's no guide RNA to target that Cas9. But as soon as I add the guide RNA expression cassette, we see a significant increase. Now, again, none of this requires prior engineering of the Candida auris strain. And so our question is, can we use these same tools to improve transformation targeted efficiency in members of diverse genetic backgrounds? And in fact, that is in what we see, that our system significantly improves the targeted integration rate in representative isolates from very diverse genetic backgrounds. And what I hope you'll appreciate here, for instance, if you want to be performing experiments with the clinical isolate from this particular clade, for instance, your chances of actually performing the targeted and intended genetic manipulation without such a system are sitting around 1% of all of your clones getting the correct mutation. But our system, gives you a fighting chance by really increasing that number about 25-fold. So using this, one more thing about this is that, as has been reported in related fungal species, this sort of system is kind of interesting in that the Candida auris cells tend to not hold on to the Cas9 into their genomes. We don't see stable integration in the majority of transformants. And so this is important because we don't want to deal with any potential off-target effects from continued constitutive Cas9 expression. And so after we've identified a clone of interest in our sample, we can just confirm that Cas9 has not been stably integrated. And this is relatively straightforward and simple to do, just an additional step in performing the mutation. So using this system, we've been able to come back and perform some targeted deletions of members of our screen. And we see that full deletion of these members of the RAM pathway, ACE2 and TAU3, recapitulate the aggregating phenotype that we mentioned before. When we come back and complement those strains, we see recapitulation of the wild type phenotype. So we've rescued the wild type morphology. And I'll mention for those interested that for this complementation, we also developed a new dominant selectable marker for use in Candida auris by codon optimizing G418. So this, I believe, was the first time that this has been used for Candida auris. So again, trying to expand the available tool set that can be used to perform molecular biology in this organism. Now, if the hypothesis here is that the aggregating morphology is due to a dysregulation of septum degrading enzymes such as CTS1, we wanted to test whether that was actually happening in our mutant strains. And indeed, what we see for the chitinase gene CTS1, we see a significant downregulation in the ACE2 and TAU3 mutant strains. Because we also know that the opposite can disrupt this process too, we checked whether the chitin synthase gene CHS2, which was another hit on our screen, was dysregulated in these strains, and it was not, in fact. Now, this was interesting to us because we were able to mine some data that's publicly available about a naturally occurring aggregating isolate. And based on publicly available RNA-seq data from this, we identified that the same chitinase CTS1 gene was significantly downregulated in the aggregating strain relative to a non-aggregating wild-type counterpart, which may suggest that the true naturally occurring morphology may be driven by dysregulation of the septum-degrading genes such as CTS1. Now, I'd also like to talk about another mutation that we identified from our screen and we're able to perform follow-up mutations in, and this is in a gene called M1, which has been described pretty well in Saccharomyces, but really has very little representation in the literature for fungal pathogens. So M1 is a kinase that regulates septins and cytokinesis. And what we see when we delete this gene in Candida auris, which is a non-filamentous fungi, as I've mentioned before, is that we see very polarized pseudohyphal growth of these cells. Since we had these cells, these mutants too, we also just checked if there was dysregulation of the septum degrading enzyme, and we see the opposite effect where we have very strong upregulation of the chitinase CTS1 in these cells, and it's still unclear the role this might be playing in the polarized growth. We were interested to see that these filaments in the M1 mutant tends to grow very similarly to naturally occurring filamentous yeast, in that they formed a Spitzenkorper-like structure at the apical tip. And so you can see that this lipophilic dye FM464, the accumulation of this dye indicating the formation of vesicles coordinating the polarized growth at the tip. And this really suggests that even in the non-filamentous Candida auris fungi, has still maintained the potential to follow some aspects of filamentous growth. So we next asked, based on reports from the naturally occurring aggregating isolates, which suggested that many times these aggregating strains were deficient in virulence compared to wild-type counterparts, we asked whether our morphogenic mutants showed similarly reduced virulence in Galeria melanella, model of infection. And as you can see, for instance, with this ACE2 line survival curve in purple here compared to the wild type in blue, we do see a mild but significant attenuation of virulence. This morphogenic mutant. Similar trend for TAF3 did not reach significance in our hands. The ELM1 mutant showed the largest attenuation of virulence compared to the wild type. So ELM1 here in black compared to wild type in blue. We also wanted to ask whether there was any alteration in the susceptibility of these strains to antifungals. And what I'm showing you here is the parental strain, AR382. While many Candida auris isolates are considerably resistant to fluconazole, AR382 is, in contrast, a susceptible strain parentally. And what we were surprised to see, and I'll explain why in just a moment, was that the Tau3 mutant almost completely ablated the susceptibility of the parental strain resistance to fluconazole. And I've summarized this was the strongest effect that we see here. So I'm showing you a heat map of each of our mutants. In this case, the hotter the color corresponds to reduced susceptibility, so increased resistance. And the Tau3 mutant that I've highlighted right here showed really the strongest effect in regards to fluconazole resistance. So we see this option in the RAM pathway, where a protein that has a suspected role in the RAM pathway is contributing increased resistance to fluconazole. We know that this is independent of the transcription factor downstream, ACE2, because we don't see that same phenotype in the ACE2 mutant. We also know that this is independent of simply of the aggregating morphology, because the ACE2 mutant, again, shows the same aggregating morphology as the Tau3 mutant. So the real question is this an external function of Tau3 to the RAM pathway, or is this a function of the terminal kinase in that pathway, CBK1? That's external to ACE2. So I posed this question to Kelsey Walworth, who's an undergraduate working with me in the laboratory. Kelsey generated a deletion mutant of CBK1, and what we can see based on these dissuffusion assays from multiple different concentrations of fluconazole is that yes, indeed, the CBK1 mutant is significantly less susceptible to fluconazole than the parental wild type strain. And that data is quantified here on the right with the CBK1 mutant zone of inhibition size for the disc diffusion assay in blue and the wild type in red here. Now, why did I say that this surprised us? Well, there is a report looking at the RAM pathway and antifungal resistance in Candida albicans. Only the difference here is quite stark. What we see in the CBK1 mutant for Candida albicans is quite opposite. In fact, we see a hypersensitivity to fluconazole compared to the wild type, which is compared to in Candida auris, we saw a reduction in susceptibility to fluconazole. So we're very excited about the possibility of network rewiring of a major signaling pathway in this organism, and are really beginning to move forward to investigate, the possibility of divergent evolution between these two organisms here. Before I end, I'd just like to mention that, as I said at the beginning of the talk, like my primary goal is to drive forward the conversation about Candida auris molecular biology, and so I'd just like to point out that all of the tools that I mentioned here that have really allowed us to efficiently and easily make some of the genetic mutations that I've talked about are publicly available. You can find all the DNA sequence and products on our AdGene page at this location, and also in our recently published paper, which just came out last month in Nature Communications. With that, I'd like to offer my thanks to my lab, who's very supportive, and my mentor, and acknowledge that this work wouldn't have been possible without the good folks at CDC who curate an excellent Candida auris isolate collection, as well as some of our collaborators who shared isolates with us. And we did our sequencing through MIGS in Pittsburgh, which is a very excellent option. I recommend that to anybody doing Illumina sequencing or the like that. And thanks to all of you as well.
Thank you, Darian, for a fantastic talk and for some tools that I think will be incredibly useful for the community. So we'll take questions for both speakers at the end. So enter your questions into the chat, and now I'm going to pass it over to Liz, who will introduce our second speaker.
Thanks, Jessica. Hi, everyone. It's my pleasure to introduce Johannes Vestman, who received his Bachelor of Medical Sciences and Master of Medical Sciences and PhD from Lund University in Sweden. Since 2017, he has been a postdoctoral researcher at the Hospital for Sick Children in Toronto, Canada, where his work has been sort of supported by a prestigious EMBO fellowship. He's currently applying for faculty positions, and I think would be interested to hear in any opportunities that might be coming down from the pipeline, if you're aware of any. Today, Johannes is going to talk to us about calcium-dependent ESCRT recruitment and lysosome exocytosis, maintaining epithelial integrity during Candida albicans invasion. Thanks so much, Johannes. Take it away.
Thank you so much. Can you confirm that you can hear me and see my screen? Okay, good, awesome. Thanks for the intro and thanks for having me. Today I'm going to talk about something that we are not experts in at all. Normally we work with macrophages, so we call this the epithelial stuff. But nonetheless, I'm going to do my best. So we're talking about Candida albicans infection in oral epithelial cells here. And I'm going to give you a short intro. I don't need to introduce Candida albicans, but that's our model pathogen. It's a polymorphic fungus colonizing various mucosal membranes, including the tongue epithelium. So we're using tongue epithelium cells here. We're using the wild type strain, and we're looking at, of course, Candida lysin, one of the known causative effects of cell deaths. So throughout this story, we're using the wild type strain and the ECE1 mutant here for Candida lysin. So we're investigating basically how Candida albicans penetrates epithelial cells and how the invasion pocket is formed. And we're trying to figure out all these characteristics of the invasion pocket. And not a lot has been done on the host side of things in terms of cell biology. So what we're interested in here is discovering different types of membrane repair mechanisms, basically how the host is kind of copes with the fungal stress. And these types of repair mechanisms have been described for bacterial infections and different types of chemical and physical damage, but has not been explored for fungal pathogens. So we're talking about repair mechanisms, and two of the most known ones are, you can see in the bottom here, it's exocytosis of lysosomes. The other one is something called ESCRT-mediated plasma membrane repair. These are two very different responses, but what they have in common is that they both respond to calcium influx. So for the lysosomal exocytosis, calcium enters the cell, and the lysosomes respond by fusing and patching up the membrane. For the ESCRT machinery, it's quite different, but it also responds to calcium, and then you have recruitment of these ESCRT components that then leads to fusion of the membrane and then scission and shedding of the damaged membrane. And they both result in maintaining the membrane integrity. So we wanted to explore these types of interactions here for Candida albicans. So yeah, the overall project aim is to how can epithelial cells overcome stress imposed by growing Candida albicans hyphae, as you see in the model here. If we break these aims down a little bit, the first one is to kind of characterize the invasion pocket. We don't really know what it consists of. We just know that it's a deep pocket that is formed, but we don't know how it's formed and what it consists of. The other thing we know is that, of course, the pocket gets damaged due to Candida lysin, but you also have the mechanical pressure exerted by the hyphae itself. And this is also something we need to take into consideration. And the third thing we want to investigate is what is the epithelial cell doing to kind of reduce the stress and to overcome these stress responses. So hopefully by the end of the talk, we will know quite a lot about this mechanism. So we start here with number one. We want to characterize the invasion pocket. And there's going to be a lot of confocal images here because our lab is basically taking images of everything. But we want to distinguish the short pocket from the long pockets. We start with the short pocket here. You can see that it's zoomed in here. And what we find out is general that the short pocket is often intact. This is seen by an intact cortical actin here in green and an intact plasma membrane. The pocket does not show any evidence of remodeling because the pocket, it's negative for LAMP1, which is a marker for lysosomes. And it's also negative for PI3P, which is a marker for early endosomes. So no evidence of fusion of endosomes or lysosomes at this stage. And we found that this pocket looks very, very different from macrophages. For example, macrophages can engulf pseudo hyphae or long hyphae, forming something that looks like an invasion pocket, but it's actually a frustrated phagosome. And this frustrated phagosome, instead of having cortical actin throughout, you see a very, very thick actin cuff at the top. And you also see fusion of lysosomes, as the green stain here is in the pocket. And you also see fusion of early endosomes because it's positive for PI3P. So pockets can be very, very different depending on the cell type and how they're formed. But in general, the short pockets are intact and they consist of membrane that resembles the plasma membrane. If we now switch to the long pocket, it's very, very different. Instead of having intact plasma membrane, you see signs of damage here. And you also see evidence of fusion of lysosomes. So while the short pocket is intact, the long pocket shows evidence of membrane remodeling, damage, et cetera, et cetera. And this is damage that we want to look at in more detail. Again, here, the short pocket is intact because it has, again, PI, this is gets a little bit complicated here, but PI45P2 is a lipid that basically binds various types of cortical actin proteins that maintains cortical actin. And again, it's intact. At long pockets, as we're losing the cortical actin here, we also lose this phosphoinositides. We think that these things are related to each other. It gets a little bit complicated, but we only see this damage occurring if we can stain for Candida lysin in the pocket. So with help with the Bernie Huby's lab, we developed an antibody that we can label with a dye to actually stain for Candida lysin inside the invasion pocket. And we often see Candida lysin here in green at sites where the cortical actin is lost. In comparison, the Candida lysin knockout strain shows intact cortical actin and of course no stain of Candida lysin. So we think that this is correlated, and of course we are not the first ones to suggesting this, but for Candida lysin to cause this damage, we suspect that calcium needs to enter the cell. So by adding ionomycin, it's a calcium ionophore, we're able to again remove cortical actin by adding an ionophore that causes calcium to leak into the cell here. So we think it has to do with calcium, influx, Candida lysin, et cetera, et cetera. But we want to show where the calcium is coming from and more of the mechanism for this. So basically, we have this control peptide that is also secreted by Candida lysin, by Candida albicans, but it doesn't cause membrane damage. And when we add this peptide, nothing really happens. The PIP2 is fine and the cortical actin is just fine. Again, if we add synthetic Candida lysin, we lose the PIP2, the dye for PIP2 goes into the cytosol, and we also mess up the actin here. But this only happens if calcium is present in the culture media. So in the bottom right here, when we add Candida lysin without extracellular calcium, the cortical actin is actually fine, suggesting that for calcium to cause these types of loss of actin, you need to have calcium in the extracellular media. And in line with this data, we can also see in the bottom right here that this PIP2 probe that also shows, that associated with cortical actin and membrane damage, we lose this PIP2 probe at the pocket for the wild type strain, but not for the Candida lysin knockout strain. And calcium influx due to Candida lysin has been shown before, but we wanted to monitor this, the patterns a little bit more. So again, before Candida lysin, the cells are quite dark, and we have a calcium probe here, we add in Candida lysine, it gets much brighter. But we don't see these long calcium signals. They actually look like this. If you look in the bottom left here, that when you add Candida lysine and you measure calcium, you get these spikes where it seems that you get a calcium influx, but then it rapidly stops again, suggesting that these are oscillations rather, where calcium leaks in, but then it stops. And we have a video here in the bottom right here that when we add Candida lysine, you see these flashes and flashes. So it's very different from if calcium would be sustained high. So obviously the cells has a way of stopping the calcium transient. So what we think is happening is that calcium leaks into the invasion pocket due to Candida lysine. It recruits an enzyme that basically hydrolyzes PIP2 into IP3 and DAG, and that sustains a bit of calcium signaling here. But again, we have these flashes, and we don't really know what they mean. What we do know so far is that the damage to the invasion pocket is mediated by Candida lysine, and it's also mediated by the influx of calcium. But again, what is the purpose of these calcium oscillations? When we're looking at the epithelial membrane during these oscillations, and when we're adding Candida lysine, we see these blebbing pattern here where the epithelial membrane blebs a little bit and kind of sheds away almost. And at this time, we had no idea what this was. We wanted to investigate a little bit more, of course. So we infected with a wild type strain, and we saw the same types of blebs inside the invasion pockets. We thought that, this is a way for the epithelial cells to respond to the damage. The blebs were never visible for the Candida lysine mutant strain. And similarly, when we add synthetic Candida lysine, we also see these massive blebs. This is the X and Y plane, and this is the Z plane here. So these massive blebs here. Again, we need calcium in the culture media to observe these blebs. So they're calcium dependent. And again, if you use the control peptide, the negative peptide that doesn't cause any damage, you don't see this blebbing at all. So we thought that perhaps the bleb could be a way for the cell to kind of remove Candida lysine. And when we're staining for Candida lysine here in the top right, we can see that these blebs that are shedded from the plasma membrane, they actually are highly enriched in Candida lysine, shown here in green. And similarly, when we infect with the wild type strain, that these blebs that are formed inside the pocket, they actually all seem to contain Candida lysine here, shown in green. So we think that, perhaps the epithelial cells are trying to remove the Candida lysine by blebbing like this. But of course, we need to find a mechanism to support this data. Yeah, so this is the current hypothesis, a way to repair the damage and remove Candida lysine. And as I briefly introduced in the introduction, is this ESCRT machinery that is a way of repairing cells. And the ESCRT components in the machinery is quite complex, but it needs a few components. It needs ALG2, which is actually the protein that responds to the calcium signal. And then it needs ALIX that binds as well. And then you have a few other components here that are accumulating at the site of damage, calcium influx, the ESCRT machinery accumulates and then causes this formation of these blebs. And these blebs look very, very similar to the ones that we see. And then if you break down the ESCRT components, you have different chimps, chimp2, chimp4, chimp6. And you have these ESCRT-related proteins, ALG2 and ALIX. And ALG2, we need to remember because it's an important inducer of the ESCRT. So when we're infecting the epithelial cells again, and we're staining for these types of ESCRT components, we can see that chimp2 accumulates to the pocket. Chimp4b accumulates to the pocket. And also they not only accumulate in the pocket, but they actually accumulate where these blebs are. So we currently think that the ESCRT components are accumulating, causing this blebbing, although again, it's just association right here. The strain that cannot produce Candida lysine, we don't show any ESCRT recruitment, we don't see any blebbing formation. But again, if we add synthetic Candida lysine, we again see recruitment of the ESCRT components at the bottom of these blebs, which is very indicative. So we try to think here, is there a way for us to kind of prevent the ESCRT recruitment and to see how that affects the viability of the cell? And we know that ALG2 is the protein that, that basically causes this ESCRT shedding to occur. So we also, first of all, want to stain for ALG2. And obviously, you can see that it very nicely is recruited to long pockets, but only if Candida lysine is secreted. The ECE mutant does not show any ALG2 recruitment. And when we're quantifying this, we find a very nice pattern that ALG2 is only recruited in very, very long pockets. So again, remember what I said early on, that the short pockets are intact. So these are also mainly negative for ALG2, but the deeper the pocket goes, the more frequently do we see ALG2 on the pocket, suggesting that these are damaged pockets, but that the epithelial cell responds to that damage. We also see ALG2 accumulation under blebs when we add synthetic Candida lysine. And similarly, we see recruitment of ALIX, which is an ALG2 binding protein under when we add in Candida lysine under the blebs. So this kind of summarizes what we're thinking happening right here. We think that there's a calcium influx. There's an early response by which the epithelial cell, we observe a recruitment of ALG2 that then recruits ESCRT components that then causes this blebbing to form. And we think that it's shedding Candida lysine away from the membrane. But then due to the tightness of the pocket, these blebs are actually trapped here. And that's why we see the pocket filled with blebs. We next wanted to silence ALG2 and basically inhibit the ESCRT recruitment and measure cell viability. So we got a pretty nice silencing here in these cells. And then again, we'd add a synthetic Candida lysine, now in a little bit lower dose to optimize the conditions a little bit. And while for the control siRNA, of course, when we add Candida lysine, we see these blebs here. When we silence ALG2, no blebs were formed, suggesting that this is ESCRT mediated vesicles that were formed. And similarly, when we add Candida lysine at this very low concentration, when we form these blebs, the epithelial cells actually remain intact, which is shown here by rhodamine 1,2,3, which basically binds to mitochondria and showed polarized mitochondria, and viable cells basically have their mitochondria polarized. In contrast, when we remove ALG2, the cell does not show the mitochondria, suggesting that the cell is dead. In fact, this is the difference in membrane blebbing with and without ALG2. Similarly, we use the same concentration of Candida lysine, and now we're staining for dead cells, basically, just for the regular PI staining. And we found this low threshold concentration that with the control siRNA, we get a very, very mild cell death. Only, I think it's maybe 10% of the cells that die. But at this low concentration, if the cells lack ALG2 and if they lack the ESCRT mediated repair, there's a 3 to 4-fold increase in cell death, suggesting that ALG2 and ESCRT is required for full cell viability during infection. So this is things that happened quite early on. The blebs are formed within minutes, but we also found some later responses. So I talked a little bit about lysosome exocytosis in the beginning here. So we looked at that as well. When we add Candida lysine, again, in presence of calcium, we'd see that all the lysosomes are actually accumulating, they're actually fusing with the plasma membrane, which is indicative of repair, because all the lamp staining here is in the plasma membrane. Again, it's calcium dependent, because if we do this in absence of calcium, you still get the holes. Don't get me wrong, the calcium is not required to make the holes, but you don't get the epithelial responses. So here you still see a little bit of staining, but this is not staining on the plasma membrane. This is basically leaky plasma membrane, leaky lysosomes, and that's why we see this type of staining. So we have this type of response here, where also after Candida lysine and the calcium influx, lysosomes are responding by fusion. The The big difference here, though, is that lysosome exocytosis is much slower. It takes about 2 hours before we see a significant difference in lysosome exocytosis. It also depends on calcium, of course. Another thing that was quite interesting is that the fusion of lysosomes was not dependent on Candida lysin, because when we're also looking in the Candida lysin mutant strain, or the EC1 mutant, we also see fusion of lysosomes, but only in very long pockets here. So in short pockets, we never see lysosome exocytosis. In long pockets, we see it independent of Candida lysin. So this seems to be a later response that doesn't really depend on Candida lysin. This is kind of what it looks like in very, very deep pockets, that for the wild type strain, the pocket goes deep here. You see some evidence of lysosome fusion, but also for the EC mutant strain, you also see lysosome fusion. So it's these responses, they occur, but they're not completely correlated to Candida lysin. So this is a lot of data, and I'm not sure if you all followed it, but this is a short summary here. And the key messages is that we know that Candida albicans invasion and Candida lysin secretion triggers various epithelial responses. And the damage causes calcium influx. It causes hydrolysis of this lipid here. And when we hydrolyze this lipid, you also lose the cortical actin, which is what we find in the deep pockets. But then as a response to this, the epithelial cell induces ESCRT recruitment and exocytosis of lysosomes to repair the plasma membrane and to remove Candida lysin away from the plasma membrane. And lastly, by silencing the ESCRT machinery, we shorten the viability of Candida lysin treated cells. This was, of course, a group effort, and a lot of the heavy load was done by project students, Mabel and Anna. And this, of course, we did this in collaboration with a few people in the lab, including Michelle, did a lot of work. We also have external collaborators, Bernard Hube, Stephanie, and Julia Naglik, that contribute to this project and in many, many different ways. And if you want to read this story, it was recently published in Cell Reports just a week ago. And this is a picture, one of the few rare occasions where our lab could gather during this pandemic. And with that, I would like to thank you all for listening. I'm happy to take any questions.
Fantastic. Thank you so much, Johannes. I think we'll start with Jessica and Darian. If you have any questions for Johannes, please put them in the chat and we'll read them out.
Yeah, sounds good. Thank you both for great talks. These were really fascinating. So for Darian, first question from Matt Blanco, which is, he's wondering if there's anything interesting surrounding your choice to drive your guide RNAs with POL2, or if this was just a choice of efficiency based on available tools.
Yeah, that's a good question. So the system that we based our Cas9 expression system on and the sgRNA expression, that was originally published by Aaron Mitchell's group. They used a POL3 promoter from the SNR52 locus, and we knew from collaboration with some workers in Leah Cohen's lab that attempts to use that same locus for attempts to find like a functioning promoter from that same locus in Candida auris were not successful. I think recently another group has published something that they suggested that they'd found the correct locus for that. But for the most part, we were coming into this with the knowledge that we didn't know where that good POL3 locus was. However, we did have good transcriptional data for some really strongly expressed POL2 genes. And so rather than try to make more guesswork about uncharacterized POL3 loci in Candida auris, we decided to take an approach using something that we had characterized for this organism.
Right, and then a question from Liz. For the CBK story, do you have any insight into differences in the cell wall between Aurus and Albicans CBK mutants that might explain your differences in fluconazole sensitivity?
Yeah, this is something we're really actively working on at this point. And I think in Albicans, it's been shown that CBK1 regulates ergosterol biosynthesis, gene expression as a stress response to fluconazole, and that potentially explains some of the hypersensitivity that's involved in the deletion in C. albicans. And so I'm really interested to see if this is potentially the same mechanism, but just inverted somehow. We kind of are thinking that potentially CBK1 is somehow regulating something similar, but maybe in a different fashion in Candida auris. I don't have an answer for you right now, but I hope we get there.
I think that's a lot. All right, I'll pass it back to Liz for a few questions for Johannes.
Great, thank you so much. Okay, so we've got 2 questions for Johannes. From Kathleen Mills. She says, Tfeb overexpression has been shown to promote lysosomal exocytosis. Did you examine the effect of Tfeb on lysosomal exocytosis in the long pockets?
That's a very good question. Yeah, so Tfeb is basically like a transcription factor that causes biogenesis of new lysosomes. So since we see a depletion of lysosomes, so to say, since they're fusing with the plasma membrane, you could expect Tfeb to get activated. The other thing is that, Tfeb is activated by calcium transients in the cytosol. So definitely we would expect Tfeb to be activated at this stage. We haven't looked for it, and we haven't done any Tfeb investigations, but I would bet money on that Tfeb is activated, that Tfeb would try to restore the pool of new lysosomes in this scenario. Yeah, that's how much I can say about it.
I'm going to read the second half of Kathleen's question because it's actually something I was going to ask too. So happy to be able to save typing. Second, do you think that lysosomal exocytosis is Candida lysin independent because it's just required to provide additional membrane to the plasma membrane to deal with large hyphae? So is it more of a physical process?
Yeah. You cannot really say that it's Candida lysin independent because if you just add it to synthetic Candida lysin, it also occurs. So it's not independent of Candida lysin, but it's also necessarily not dependent on it. So we were really surprised when we saw a lysosome fusion in the EC1 mutant. I must say that at the time we thought everything would be dependent on Candida lysin. I think it kind of makes sense that very, very long pockets, you need that extra membrane to form those deep pockets. And I think also it has to do with if you have mechanical pressure by the extending hyphae, eventually you will have some breaches of the plasma membrane. And as long calcium can enter the cell, you would have responses such as this. But yeah, it's weird because we never see the blebs with the mutant, so we still don't know all of it. But yeah, I would think along those lines for sure.
And just a clarification here, for the mutants, you're talking about the full length ECE1 deletion.
Yeah, exactly. The full length deletion. And it doesn't seem to matter if we put back all the other genes except Candida lysin, we see the same effect. So, but although we haven't tested that strain for lysosome exocytosis, but nonetheless, mechanical pressure is sufficient for lysosome exocytosis if the pocket is very deep. But it occurs quite late. It takes 3 to 4 hours when we see this in the pockets.
Thanks. Go ahead, Jessica.
All right, Darian, there are several questions about regulation of filamentation in Candida auris. And so Kathleen Mills says that it's very interesting to her that only one gene seems to prevent filamentation. And are there any instances of naturally occurring deletants in that gene that resulting in filamentation?
Yeah, this is a good question. So we know from Saccharomyces that L1 regulates septins and during cytokinesis. And while we haven't experimentally confirmed that it has the same role in Candida auris, like potentially just simply disrupting the septins is sufficient to trigger a morphogenesis checkpoint, such as through SWE1, and result in such polarized growth. And so that's kind of the working hypothesis, I guess, for how disruption of just a single gene could promote this sort of phenotype. As far as your second question, yeah, there's there are examples of, naturally occurring isolated Candida auris mutants that grow in similar filamentous or polarized phenotypes, but the majority of the time there's not a genetic explanation for them. So a lot of times they've been recovered from an infection model or something like that in the literature, and then just reported as is with the phenotype, but no genetic description. And so maybe is the answer to the question. Perhaps there's some pressure against Elm1 in some of these mutants, but I think it hasn't been looked at by many of the authors who published those results. But I think that's a very interesting point, very interesting possibility.
Yeah, and Liz asked a bit more about the Elm1 dependent inhibition of filamentation, and you've answered part of it, but just to get her question on the record, she asked whether you think that Elm1 dependent inhibition of filamentation is just condition dependent, or is there a more stringent mechanism?
Yeah, as I was saying, I think it probably has to do with disruption of septin. And it seems, so for our mutants, for instance, it's a constitutive process. Like this is not a response in this case. So I think it's just probably a disruption of the growth phase of these organisms.
And I was very curious about the Spitzenkorper-like structure that you see. And are the genes involved in formation of those structures well conserved across the different clades of Candida auris, even though they don't seem to be expressed and produced, or have they been lost in some groups?
Yeah, this is a really interesting question. I haven't done the analysis across different clades. What I can tell you is that, many of the, you see formin and polarosome genes that have good orthologs in Candida auris. Most of the, I've really only looked at clade 1. That's where we've done most of the genetic work. And so I don't know for sure whether they're expressed in, whether they're encoded by the other clades too. We haven't looked at their expression either. So that's really interesting. That would be really interesting to see, and probably one of our next steps in trying to analyze this M1 mutant is to see if these cells are actually turning on, similarly Spitzenkorper related genes.
Thank you very much. Liz, do you want to finish off?
Yeah, we've got a whole bunch of questions coming through. I'm going to try and group them here. So Johannes, Julian Naglith has asked, is the ESCRT-dependent epithelial repair response to Candida lysin similar to other bacterial toxins, or are there Candida lysin specific aspects?
As far as we know, they're very similar, although we haven't done any studies on that, but I think so. They're very similar.
So Matthew Blango has asked, are the blebs coming from the plasma membrane, multiple vesicular bodies? He's sorry if he missed this, if you mentioned it.
Yeah, so the blebs are basically when the damaged plasma membrane is pinched together, and then you basically get the shedding and scission of damaged plasma membrane. Okay.
Lilia says, wonderful work. Is the recycling endosomal mechanism involved in the lysosome exocytosis, or how lysosome exocytosis happens?
To my knowledge, it's not involved. I don't think there are any recycling mechanisms involved, as far as I know. I can be wrong. And in terms of how lysosomes are fusing with the membrane, I would say we don't really know more than that lysosomes are migrating on microtubule network. And I think that complex have involved proteins that are calcium sensitive. So I think basically they are, the lysosomes, they migrate on mitochondria and they can sense calcium transients, and they would just move in that direction and then fuse with their target, whatever that is.
Okay. So Kelly Shepherdson asks, very interesting data on how damage is mitigated by the host cell, because you see breakdown of F-actin in the cells following Candida lysin. I'm wondering if you think fragments of F-actin are potentially leaking from the cell, and whether this has any additional effect on the responses on the cell, acting as a damp, for example, and maybe an additional signal potentially related to the lysosomes.
That's also a great question. It has to do with the size of the fragments. If the actin fragments are small enough, I'm sure they could leak. Basically, the invasion pocket, the top of the invasion pocket is quite tight. If you add antibodies to stain for proteins inside the pockets, antibodies are too large to enter. So there's some type of cutoff there. While like 10 kilodalton dextran can go in, and some fluorescent dyes can go in. So I think if the F-actin fragments are small enough, they could definitely leak out. I think basically G-actin monomers could leak out. So I would expect some of that to happen, and I would expect them to act as a damp for sure.
Really interesting. Okay, so Julian Nadlick says, is there any evidence that the PTDINs, other than the 4,5P2 are involved in epithelial repair response to Candida lysis?
Not to my knowledge. I think that is the main signal, so loss of PIP2. So yeah, I guess you could call it a part of the mechanism that the loss of PIP2 causes IP3, which is, goes to the ER and it releases more calcium. And to my knowledge, no other phosphonostatides are involved, but I might be wrong. Maybe someone shows that in the future.
Okay, great. And last one from Kevin Newhall, he says, great talk. The increased death you observed from your ALG2-KD experiments, were you able to determine the kind of cell death? Was there any inflammasome formation or LB1B maturation in these cells?
Yeah, no, we could not distinguish what type of cell death. We just did DAPI staining, propydium iodide staining, calling it, general necrosis. I'm gonna guess, but I don't know. I'm not an expert in this. We did not measure inflammasome activation or IL-1 beta cleavage or secretion. I think a lot of the cell death responses occur so fast. So when we add in candidalysin in this type of kind of intermediate concentration, I think we measured cell death after 1 hour. So at that time, these cells are not primed in any way with LPS or whatever. So I don't think that you would have, first transcription of the right genes and then the caspase activation and secretion in cell death in that way. So I'm gonna guess it's not related to inflammasome activation, but we haven't tested it.
All right, that's all the questions in the chat. If anybody wants to turn on their camera and ask a follow-up question, I think we can do that. I don't see any hands. Jessica, you want to close us out?
Sounds good. Thanks everyone for coming. And we're looking for more speakers. So if you're at all interested, please either reach out or submit something through our website. Or if you know someone who you think would be a great speaker, reach out and we're happy to then send out invitations to them. So we hope to see you in February, and we also hope to hear from you about your science. Thanks everyone.